Good morning, students. Students, as in our previous video, we were discussing about the applications of biotechnology in the field of medicines. And in our previous video, we discussed about the biotechnologically synthesized insulin. In the same series today, I will discuss about a new application, and the application is gene therapy. Now, as name is suggesting, gene therapy is a kind of therapy which is used to correct or to treat the hereditary diseases. Means if there is any defect in the gene and a particular gene of human being is not functioning properly, then if it is diagnosed in the early lifespan, or I have to say that if that particular gene which is defected or which is not functional has been detected in the early stage of the life cycle or in the childhood or during the embryonic stage, then we can correct or we can use this gene therapy, and we can convert this non-functional gene into a functional gene. Now, what are the methods of this therapy or gene therapy? I will tell you. Now, it involves basically in this method, the correct gene, or I have to say that the functional genes are inserted into a cells or a particular tissue to treat that particular disease. So what we have to do here, as an individual is having a non-functional gene, if we are going to diagnose it in the early stages of the life cycle or in the childhood, or we have to say that during the embryonic development, if we are going to identify or we are going to diagnose that that particular gene is not functioning properly, so what can we do? We can introduce a functional gene in the DNA of that particular child, or in that particular embryo. So the non-functional gene will not function. So definitely, if there will be there will be an additional gene, that additional gene will be functional, or the recombinant DNA which is having that functional gene will synthesize mRNA during the transcription, and that mRNA will participate in the translation process and will produce that particular product which is required, and we can treat that particular disease. Now I will tell you that what is the principle of gene therapy. Normal gene, when get inserted into the individual cell or embryo with gene defect, the normal gene will take over the function and compensate the non-functional gene. So as I mentioned earlier, that if there is a person, or there is a child, or there is an embryo uh, which has a gene, and that particular gene is not functioning properly, so what we have to do, we have to add one additional gene, or we have to say that we have to add one functional gene in that particular cell of the individual, or in that particular tissue which is functional, and this functional gene will compensate the function of non-functional gene. And I want to tell you that the first gene therapy was done in 1990, uh, and it was performed in a 4-year-old girl child, and she was suffering from a disease, and the name of the disease is adenosine deaminase deficiency disease, and in short it is called ADA. A for adenosine, D for deaminase, A for deaminase and deficiency disease. ठीक है, and this disease can be treated in the earlier stage of the life cycle. What we can do in the early stages of the life cycle, ठीक है, in the early stages bone marrow transplantation can occur, ठीक है, by enzyme replacement theory. तो in early stages of the life cycle, where all the cells are going to be formed, especially the blood cells, if we are going to transplant the bone marrow, that then this disease can be cured. Now uh, I will tell you some other biotechnological methods which can be used to treat that particular disease. ठीक है, ADA deficiency यानी adenosine deaminase deficiency disease can be also treated. It is a hereditary disease which passes from parents to their offspring, and this hereditary disease or disorder can be cured by deletion of that non-functional adenosine deaminase gene. So it means what we have to do here, ठीक है, there is a gene, that particular gene is participating in the transcription and forming one mRNA, and this mRNA is coding for a protein, and this protein is especially a kind of enzyme, and the name of this enzyme is adenosine deaminase enzyme. ठीक है, uh, and this enzyme is very important, and this enzyme plays a very important role in our immune system function. ठीक है, while we are going to treat this disease, what we have to do in gene therapy, the lymphocyte of a patient are taken out. ठीक है, what we have to do, हमें किसी patient से lymphocytes को एक्सट्रैक्ट करना है, उससे उसके lymphocytes को निकालना है. We have to isolate the lymphocytes from the blood of the patient, and we have to grow them into a culture medium, and artificial एक culture medium हमें बनाना है, and we have to grow these lymphocytes into that particular artificial medium. And what we have to do now, फंक्शनल एडीए cDNA मीन्स एक ऐसा डीएनए जिसको हम बोल सकते हैं कि एक रिकॉम्बिनेंट डीएनए है, इसको हम बोलते हैं फंक्शनल एडीए cDNA, मतलब एक ऐसा रिकॉम्बिनेंट डीएनए जिसके पास कि इस एंजाइम को बनाने वाला जीन प्रेजेंट है. ठीक है, अब फंक्शनल एडीए cDNA इज इंट्रोड्यूस इनटू लिम्फोसाइट्स. तो अब जो हमने लिम्फोसाइट कल्चर मीडियम में कल्चर की हुई हैं, now we have to, what we have to do, we have to add that particular gene in the DNA of those lymphocytes. ठीक है, and how can we introduce, we have to use some vectors, and here as a vector we are using retroviruses. ठीक है, and we are using retroviruses as a vector. Initially we are inserting those functional gene into the retroviruses, and then we have to uh, infect the lymphocyte from these retroviruses, and when they will infect the lymphocytes, definitely they will release the ADA cDNA into the lymphocytes. now अब जो lymphocyte culture medium में अब उनके पास वो जीन आ चुका है जो आपका ADA एंजाइम बनाता है. नाउ ठीक है, नाउ सिंस द लिम्फोसाइट्स हैव शॉर्ट लाइफ स्पान, अब हम क्या करेंगे, उन लिम्फोसाइट्स को दोबारा पेशेंट की बॉडी में डाल देंगे, और पेशेंट की बॉडी में आपका ये एडीए जो एंजाइम है ये बनना स्टार्ट हो जाएगा. नाउ सिंस लिम्फोसाइट्स हैव लाइफ शॉर्ट लाइफ स्पान, तो एज वी नो दैट लिम्फोसाइट्स का जो लाइफ स्पान है वेरी शॉर्ट है. ठीक है, तो जो जेनेटिकली इंजीनियर्ड आपके लिम्फोसाइट्स थे, ये एक फिक्स पीरियड के बाद ये इनकी डेथ होगी, तो दोबारा डिफिशियंसी हो जाएगी. तो पीरियडिकली वी हैव टू इंट्रोड्यूस दिस काइंड ऑफ लिम्फोसाइट इनटू अ पेशेंट body. But if we are going to diagnose that a particular individual is suffering from that particular disease, तो ठीक है, तो what we can do, we can in the during the early embryonic stage or during the early childhood, these genes can be isolated from the bone marrow cell, and we can introduce a new kind of cell into the bone marrow, and this is the permanent cure, and this is done only in the early embryonic stage. ठीक है, but if gene isolate from bone marrow cells producing ADA introduced into the cells of the embryonic stage, uh, then it's a permanent cure. तो what we have to do here, तो अगर हमने अर्ली एम्ब्रियोनिक स्टेज पे डायग्नोस कर लिया, क्योंकि अर्ली एम्ब्रियोनिक स्टेज पे अभी बहुत सारे ऑर्गन आपके मैच्योर नहीं हुए हैं, इन दैट पर्टिकुलर स्टेज हमको क्या करना है, किसी फंक्शनल बोन मैरो से, मीन्स एक ऐसा पर्सन जो कि आपका नॉर्मल पर्सन है जिसमें एडीए प्रॉपरली बनता है, तो उसके बोन मैरो से आपने सेल को आइसोलेट करना है और उसे इंजेक्ट कर देना है डीएनए को, कहाँ, एक एम्ब्रियो के पास जिसके पास की नॉर्मल जीन नहीं है. ठीक है, हेयर इन द पिक्चर यू कैन सी दैट what is the procedure. Now cells are harvested from the patient. Now in lab they are cultured into a artificial culture medium, and here uh, in that particular artificial medium, now the genes are then introduced into the cells through viruses, and the viruses are releasing their genetic material into the lymphocytes, and these lymphocytes are becoming uh, transgenic. I have to say that these cells are genetically altered or genetically modified, and finally these genetically modified cells are once again introduced into the human body. So this is the concept of gene therapy. Okay, I hope so you understood the concept of gene therapy. Now along with it I want to discuss some few more new topics, and this topic is molecular diagnostic. Molecular diagnostic means if we are going to identify is there any defect in the molecular analogs, means is there any defect in the DNA, and we are going to diagnose it and we are going to treat it. ठीक है, now molecular diagnostic techniques are used to diagnose or monitor disease, detect risk, and decide which therapies will work best for the individual patient. Now now these techniques are generally used for the diagnosis of particular disease. If there is an hereditary disease, there is a hereditary disease, and so with the help of molecular diagnostic we can identify is there any gene in the DNA which is defected. So we are going to identify, or we are going to identify how much risk is there due to that particular gene, and we can decide that which can technique we can use to treat that particular disease. ठीक है. Treatments of a disease be effective. Early diagnosis and understanding the pathophysiology is very important. So it's very important that we are going to detect that particular gene which is defected in the early stages of the life cycle. So as early as we will diagnose that particular defected gene, so we can understood its pathophysiology and we can go for the treatment. ठीक है. The conventional methods which are used to detect or to diagnose any disorder are urine test or serum test. ठीक है. But with the help of these tests, early detection of that particular disease is not possible. So for early detection, what can we do? For early detection of that particular disease, we can go for rDNA technology, or we can use PCR, polymerase chain reaction, or we can go for ELISA, enzyme linked immunosorbent assay. ठीक है. And there are some conventional methods that detect presence of pathogens, bacteria, viruses. When disease is well established and produce diseases symptom pathogen high body means कन्वेंशनल मेथड्स में हम क्या करते हैं, हम जनरली हम बॉडी को ब्लड सैंपल ले लेते हैं, यूरिन सैंपल ले लेते हैं, और हम फाइंड आउट करने की कोशिश करते हैं कि क्या इन सैंपल में कोई इन्फेक्शियस एजेंट, बैक्टीरिया या वायरसेस प्रेजेंट है या नहीं है. बट इट विल टेक अ लॉन्गर टाइम. तब तक डिजीज जो होगी पूरी तरह से इस्टेब्लिश हो चुकी होगी और उस डिजीज़ को क्योर करना थोड़ा सा मुश्किल होगा. सो वी हैव टू गो फॉर द अर्ली डायग्नोसिस methods like rDNA technology, PCR और ELISA. ठीक है. नाउ व्हाट इज़ द सिग्निफिकेंस ऑफ मॉलिक्यूलर डायग्नोस्टिक, क्या फ़ायदे हैं इसके, वी आर गोइंग टू डिस्कस अबाउट इट. द मॉलिक्यूलर डायग्नोस्टिक्स लाइक PCR डिटेक्ट्स पैथोजेंस इवन एट वेरी लो कंसंट्रेशन. तो अगर ब्लड में बहुत कम अमाउंट में भी वायरसेस हैं तो विद द हेल्प ऑफ द PCR, ठीक है, बहुत कम वायरसेस हैं तो आपके जो सेल्स होंगी उतनी ज़्यादा डैमेज नहीं हो रही होंगी. तो आप क्या करेंगे, PCR टेक्निक्स की मदद से जो आपका DNA है उसको कई सा बार एम्पलीफाई करके उसकी स्टडी कर सकते हैं. एंड यू कैन आइडेंटिफाई इज़ देयर एनी डिफेक्ट इन द DNA, और अर्ली स्टेजेस में जब वायरसेस का नंबर कम है आप उसे डिटेक्ट कर सकते हैं. ठीक है. एंड इफ़ यू आर second, we I am giving you an example. If there is a person and a person is suspected that he might be it might be possible that he is having HIV. तो हम खाली, अगर हमें सस्पेक्टेड पर्सन है, अगर जब तक वो उसमें सिम्टम आएंगे तब जाके उसको क्योर करने जाएंगे तो काफ़ी टाइम लग जाएगा. इफ़ सस्पेक्टेड पर्सन का अगर हम पहले से ही हम उसका बायोटेक्नोलॉजिकल मेथड से अगर हम उसका डायग्नोस करने की कोशिश करेंगे तो वी कैन प्रिवेंट हिम फ्रॉम दैट पर्टिकुलर डिजीज़. ठीक है. नाउ सिमिलरली वी कैन फाइंड आउट द म्यूटेटेड जीन्स टू. ठीक है. वी कैन फाइंड आउट एनी इफ देयर एनी जेनेटिक डिसऑर्डर टू. ठीक है. एंड इन दैट पर्टिकुलर टेक्निक वी, while we are going for the molecular diagnosis, we have to use PCR. And PCR, what as we know that what is the gene for the normal person and what is a defected gene, so we can use a hybrid complementary DNA and we can find out after the autoradiography we can find out that is that particular gene is mutated or non-mutated or is a normal gene. So with the help of molecular diagnostic techniques we can easily find out that particular disease. There is one more test which is called ELISA. ELISA is a diagnostic method to detect antigens or foreign substance or pathogen. Means ELISA is enzyme linked immunosorbent assay, in which in this test basically this test works on a principle antigen antibody interaction. If there will be any antigen, so in our body definitely some kind of antibodies will be produced. ठीक है. So special kind of protein is there, special kind of glycoprotein is there, special kind of antigen is there. For it our body will produce antibodies and we have to identify these antigen or antibodies in our body, and we can find out that is there any chance of infection from any particular virus or bacteria. So I am concluding myself right now. ठीक है. I hope so you understood the concept of gene therapy as well as the molecular diagnostic. Once again I will meet you in the next class. Till then, thank you, bye bye, and have a nice day.